In this video, we will be covering Gibson assembly. Gibson assembly is a cloning technique that joins multiple DNA fragments together in an isothermal reaction. Advantages with Gibson assembly include its cheap and fast cloning scheme, minimal steps, and ability to combine up to 5 DNA fragments with a one-step master mix of enzymes. Without further ado, let's begin. Similar to other DNA assembly methods, the initial step requires one to determine the DNA fragments that they wish to assemble. Once that is completed, each DNA fragment must be amplified through the use of PCR. As a reminder, PCR is completed by splitting the DNA strand into 2 strands and the RNA polymerase coming in and attaching a complementary nucleotides on each side. Once the targeted genes are amplified, combine equal molar amounts of the DNA fragments into a single reaction tube with a master mix. This master mix contains 3 enzymes: T5-exonuclease, DNA polymerase, and tag DNA ligase. Once the DNA fragments and the master mix are in the same tube, they must be incubated for 1 hour. During this incubation period, the T5-exonuclease selectively removes nucleotides from the 5' ends of DNA fragments to ensure that DNA fragments overhangs are compatible with one another. Then, the DNA polymerase anneals the DNA fragments together and fills in any gaps with nucleotides. Lastly, DNA ligase completes the Gibson assembly method by sealing the DNA fragments together and ensuring that a continuous and intact DNA molecule is built. Using Gibson assembly, we just combined 2 DNA fragments. From there, the DNA molecule created can be inserted into a host organism, such as bacteria, and put to use.